electron micrograph shows a long, thin chromosome that has a banding pattern. Figure 13. 1 chromosomes are thread-like nuclear structures consisting of DNA and proteins that serve as the repositories for genetic information. The chromosomes depicted here were isolated from a fruit fly's salivary gland, stained with dye, and visualized under a microscope. Akin to miniature barcodes, chromosomes absorb different dyes to produce characteristic banding patterns, which allows for their routine identification. Credit modification of work by LPLT/Wikimedia Commons. Scale bar data from Matt Russell. Chapter outline. 13. 1 chromosomal theory and genetic linkage. 13. 2 chromosomal basis of inherited disorders. The gene is the physical unit of inheritance, and genes are arranged in a linear order on chromosomes. Chromosome behavior and interaction during meiosis explain, at a cellular level, inheritance patterns that we observe in populations. Genetic disorders involving alterations in chromosome number or structure may have dramatic effects and can prevent a fertilized egg from developing. Learning objectives. By the end of this section, you will be able to do the following. Discuss Sutton's chromosomal theory of inheritance. Describe genetic linkage. Explain the process of homologous recombination, or crossing over. Describe chromosome creation. Calculate the distances between 3 genes on a chromosome using a 3-point test cross. Long before scientists visualized chromosomes under a microscope, the father of modern genetics, Gregor Mendel, began studying heredity in 1843. With improved microscopic techniques during the late 1800s, cell biologists could stain and visualize subcellular structures with dyes and observe their actions during cell division and meiosis. With each mitotic division, chromosomes replicated, condensed from an amorphous, no constant shape, nuclear mass into distinct X-shaped bodies, pairs of identical sister chromatids, and migrated to separate cellular poles. Chromosomal theory of inheritance. The speculation that chromosomes might be the key to understanding heredity led several scientists to examine Mendel's publications and reevaluate his model in terms of chromosome behavior during mitosis and meiosis. In 1902, Theodore Boveri observed that proper sea urchin embryonic development does not occur unless chromosomes are present. That same year, Walter Sutton observed chromosome separation into daughter cells during meiosis, Figure 13. 2. Together, these observations led to the chromosomal theory of inheritance, which identified chromosomes as the genetic material responsible for Mendelian inheritance. Part A is a photo of Walter Sutton. Part B is a photo of Theodore Boveri. Part C is a picture of Aline Carruthers holding a grasshopper cage. Figure 13.2. A. Walter Sutton and B. Theodore Boveri developed the chromosomal theory of inheritance, which states that chromosomes carry the unit of heredity, genes. Aline Carruthers, C., was the first to provide physical evidence supporting the theory. The chromosomal theory of inheritance was consistent with Mendel's laws, which the following observations supported. During meiosis, homologous chromosome pairs migrate as discrete structures that are independent of other chromosome pairs. Chromosome sorting from each homologous pair into pregametes appears to be random. Each parent synthesizes gametes that contain only half their chromosomal complement. Even though male and female gametes, sperm and egg, differ in size and morphology, they have the same number of chromosomes, suggesting equal genetic contributions from each parent. The gametic chromosomes combine during fertilization to produce offspring with the same chromosome number as their parents. Despite the lack of direct evidence that chromosomes carry traits, the compelling correlation between chromosome behavior during meiosis and Mendel's abstract laws led scientists to propose the chromosomal theory of inheritance. Critics pointed out that individuals had far more independently segregating traits than they had chromosomes. About 10 years after the theory was proposed, Aline Carruthers was the first to discover physical evidence supporting it. She observed independent chromosome assortment in grasshoppers. Then, after several years of carrying out crosses with the fruit fly, Drosophila melanogaster, Thomas Hunt Morgan provided additional experimental evidence to support the chromosomal theory of inheritance. Genetic linkage and distances. Mendel's work suggested that traits are inherited independently of each other. Morgan identified a 1:1 correspondence between a segregating trait and the X chromosome, suggesting that random chromosome segregation was the physical basis of Mendel's model. This also demonstrated that linked genes disrupt Mendel's predicted outcomes. That each chromosome can carry many linked genes explains how individuals can have many more traits than they have chromosomes. However, researchers in Morgan's laboratory suggested that alleles positioned on the same chromosome were not always inherited together. During meiosis, linked genes somehow became unlinked. Homologous recombination. In 1909, Franz Janssen observed chiasmata, the point at which chromatids are in contact with each other and may exchange segments, prior to the first meiotic division. He suggested that alleles become unlinked and chromosomes physically exchange segments. As chromosomes condensed and paired with their homologs, they appeared to interact at distinct points. Janssen suggested that these points corresponded to regions in which chromosome segments exchanged. We now know that the pairing and interaction between homologous chromosomes, or synapsis, does more than simply organize the homologs for migration to separate daughter cells. When synapsed, homologous chromosomes undergo reciprocal physical exchanges at their arms in homologous recombination, or more simply, crossing over. To better understand the type of experimental results that researchers were obtaining at this time, consider a heterozygous individual that inherited dominant maternal alleles for two genes on the same chromosome, such as A and B, and two recessive paternal alleles for those same genes, such as A and B. If the genes are linked, one would expect this individual to produce gametes that are either OB or OB with a 1:1 ratio. If the genes are unlinked, the individual should produce OB, OB, AB, and OB gametes with equal frequencies, according to the Mendelian concept of independent assortment. Because they correspond to new allele combinations, the genotypes OB and AB are nonparental types that result from homologous recombination during meiosis. Parental types are progeny that exhibit the same allelic combination as their parents. Morgan and his colleagues, however, found that when they test-crossed such heterozygous individuals to a homozygous recessive parent, OBB times OB, both parental and nonparental cases occurred. For example, 950 offspring might be recovered that were either OBB or OB, but 50 offspring would also result that were either OB or OBB. These results suggested that linkage occurred most often, but a significant minority of offspring were the products of recombination. One of the experiments in Morgan's lab involving the crosses of flies for two traits, body color, gray or black, and wing shape, normal and vestigial, demonstrated the recombination events that lead to the development of nonparental phenotypes, Figure 13. 3. Visual connection. The illustration shows the possible inheritance patterns of linked and unlinked genes. The example used includes fruit fly body color and wing length. Fruit flies may have a dominant gray color lowercase b superscript plus or a recessive black color lowercase b. They may have dominant long wings lowercase vg superscript plus or recessive short wings lowercase vg. 3 hypothetical inheritance patterns for a test cross between a heterozygous and a recessive fruit fly are shown, based on gene placement. The actual experimental results published by Thomas Hunt Morgan in 1912 are also shown. In the first hypothetical inheritance pattern in part A, the genes for the two characteristics are on different chromosomes. Independent assortment occurs so that the ratio of genotypes in the offspring is 1b plus bvg plus Vg colon, 1bb Vg Vg colon, 1b plus bvg Vg colon, 1bb Vg plus Vg, and 50% of the offspring are nonparental types. In the second hypothetical inheritance pattern in Part B, the genes are close together on the same chromosome so that no crossover occurs between them. The ratio of genotypes is 1b plus bvg plus Vg colon 1, bbvgvg, and none of the offspring are recombinant. In the third hypothetical inheritance pattern in Part C, the genes are far apart on the same chromosome so that crossing over occurs 100% of the time. The ratio of genotypes is the same as for genes on two different chromosomes, and 50% of the offspring are recombinant, nonparental types. Part D shows that the number of offspring that Thomas Hunt Morgan actually observed was 965:944:206:185, which is B plus BVG plus VG colon B BVG VG colon B plus BVG VG colon B BVG plus VG. 17% of the offspring were recombinant, indicating that the genes are on the same chromosome and crossing over occurs between them some of the time. Figure 13. 3. This figure shows unlinked and linked gene inheritance patterns. The example used includes fruit fly body color and wing length. Fruit flies may have a dominant gray color B plus or a recessive black color B. They may have dominant long wings VG plus or recessive short wings VG. In A, 2 genes are located on different chromosomes, so independent assortment occurs during meiosis. The offspring have an equal chance of being the parental type, inheriting the same combination of traits as the parents, or a non-parental type, inheriting a different combination of traits than the parents. In B, 2 genes are very close together on the same chromosome so that no crossing over occurs between them. Therefore, the genes are always inherited together, and all the offspring are the parental type. In C, 2 genes are far apart on the chromosome such that crossing over occurs during every meiotic event. The recombination frequency will be the same as if the genes were on separate chromosomes. D, the actual recombination frequency of fruit fly wing length and body color that Thomas Morgan observed in 1912 was 17%. A crossover frequency between 0% and 50% indicates that the genes are on the same chromosome and crossover sometimes occurs. In a test cross for 2 characteristics such as the one here, can the recombinant offspring's predicted frequency be 60%? Why or why not? Genetic maps. Janssen did not have the technology to demonstrate crossing over, so it remained an abstract idea that scientists did not widely believe. Scientists thought chiasmata were a variation on synapsis and could not understand how chromosomes could break and rejoin. Yet, the data were clear that linkage did not always occur. Ultimately, it took a young undergraduate student and an all-nighter to mathematically elucidate the linkage and recombination problem. In 1913, Alfred Sturtevant, a student in Morgan's laboratory, gathered results from researchers in the laboratory and took them home one night to mull them over. By the next morning, he had created the first chromosome map, a linear representation of gene order and relative distance on a chromosome, figure 13.4. Visual connection. The illustration shows a Drosophila genetic map. The gene for aristide length occurs at 0 centimorgans, or lowercase c uppercase m. The gene for body color occurs at 48. 5 lower c upper m. The gene for red versus cinnabar eye color occurs at 57. 5 lower c upper m. The gene for wing length occurs at 65. 5 lower c upper m. And the gene for red versus brown eye color occurs at 104. 5 lower c upper m, 1 lower c upper m is equivalent to a recombination frequency of 0. 1. Figure 13. For this genetic map orders Drosophila genes on the basis of recombination frequency. Which of the following statements is true? Recombination of the body color and red cinnabar eye alleles will occur more frequently than recombination of the alleles for wing length and aristide length. Recombination of the body color and aristide length alleles will occur more frequently than recombination of red-brown eye alleles and the aristide length alleles. Recombination of the gray-black body color and long-short aristide alleles will not occur. Recombination of the red-brown eye and long-short aristide alleles will occur more frequently than recombination of the alleles for wing length and body color, as figure 13. 4 shows, by using recombination frequency to predict genetic distance, we can infer the relative gene order on chromosome 2. The values represent map distances in centimorgans, cm, which correspond to recombination frequencies, in percent. Therefore, the genes for body color and wing size were 65 5 minus 48. 5 equals 17 cm apart, indicating that the maternal and paternal alleles for these genes recombine in 17% of offspring, on average. To construct a chromosome map, Sturtevant assumed that genes were ordered serially on thread-like chromosomes. He also assumed that the incidence of recombination between two homologous chromosomes could occur with equal likelihood anywhere along the chromosome's length. Operating under these assumptions, Sturtevant postulated that alleles that were far apart on a chromosome were more likely to dissociate during meiosis, simply because there was a larger region over which recombination could occur. Conversely, alleles that were close to each other on the chromosome were likely to be inherited together. The average number of crossovers between two alleles, that is, their recombination frequency, correlated with their genetic distance from each other, relative to the locations of other genes on that chromosome. Considering the example cross between OB and OB above, we could calculate the recombination's frequency as 51,000 equals 0. 5. That is, the likelihood of a crossover between genes AA and BB was 0.5, or 5%. Such a result would indicate that the genes were definitively linked, but that they were far enough apart for crossovers to occasionally occur. Sturtevant divided his genetic map into map units, or centimorgans, cm, in which a 0-1 recombination frequency corresponds to 1 cm. By representing alleles in a linear map, Sturtevant suggested that genes can range from linking perfectly, recombination frequency equals 0, to unlinking perfectly, recombination frequency equals 0. 5. When genes are on different chromosomes, or genes separate very far apart on the same chromosome. Perfectly unlinked genes correspond to the frequencies Mendel predicted to assort independently in a dihybrid cross. A 0.5 recombination frequency indicates that 50% of offspring are recombinants and the other 50% are parental types. That is, every type of allele combination is represented with equal frequency. This representation allowed Sturtevant to additively calculate distances between several genes on the same chromosome. However, as the genetic distances approached 0.50, his predictions became less accurate because it was not clear whether the genes were very far apart on the same or on different chromosomes. In 1931, Barbara McClintock and Harriet Creighton demonstrated the crossover of homologous chromosomes in corn plants. Weeks later, Kurt Stern demonstrated microscopically homologous recombination in Drosophila. Stern observed several X-linked phenotypes that were associated with a structurally unusual and dissimilar X chromosome pair in which one X was missing a small terminal segment, and the other X was fused to a piece of the Y chromosome. By crossing flies, observing their offspring, and then visualizing the offspring's chromosomes, Stern demonstrated that every time the offspring allele combination deviated from either of the parental combinations, there was a corresponding exchange of an X chromosome segment. Using mutant flies with structurally distinct X chromosomes was the key to observing the products of recombination, because DNA sequencing and other molecular tools were not yet available. We now know that homologous chromosomes regularly exchange segments in meiosis by reciprocally breaking and rejoining their DNA at precise locations. Aurora Ruiz Herrera, for example, studies the occurrence of genetic breakpoints at locations in the chromosomes known as fragile sites. By identifying chromosomal fragile sites that are shared between humans and other primates, Ruiz Herrera has provided a deeper understanding of mammalian and specifically human evolution. Link to learning. Review Sturtevant's process to create a genetic map on the basis of recombination frequencies here. Mendel's mapped traits. Homologous recombination is a common genetic process, yet Mendel never observed it. Had he investigated both linked and unlinked genes, it would have been much more difficult for him to create a unified model of his data on the basis of probabilistic calculations. Researchers who have since mapped the 7 traits that Mendel investigated onto a pea plant genomes, 7 chromosomes, have confirmed that all the genes he examined are either on separate chromosomes or are sufficiently far apart as to be statistically unlinked. Some have suggested that Mendel was enormously lucky to select only unlinked genes, whereas others question whether Mendel discarded any data suggesting linkage. In any case, Mendel consistently observed independent assortment because he examined genes that were effectively unlinked. Learning objectives. By the end of this section, you will be able to do the following. Describe how a karyogram is created. Explain how non-disjunction leads to disorders in chromosome number. Compare disorders that aneuploidy causes. Describe how errors in chromosome structure occur through inversions and translocations. Inherited disorders can arise when chromosomes behave abnormally during meiosis. We can divide chromosome disorders into two categories: abnormalities in chromosome number and chromosomal structural rearrangements. Because even small chromosome segments can span many genes, chromosomal disorders are characteristically dramatic and often fatal. Chromosome identification. Chromosome isolation and microscopic observation forms the basis of cytogenetics and is the primary method by which clinicians detect chromosomal abnormalities in humans. A karyotype is the number and appearance of chromosomes and includes their length, banding pattern, and centromere position. To obtain a view of an individual's karyotype, cytologists photograph the chromosomes and then cut and paste each chromosome into a chart or karyogram. Another name is an ideogram, figure 13.5. This is a karyotype of a human female. There are 22 homologous pairs of chromosomes and an X-sex chromosome pair. Figure 13.5. This karyotype is of a female human. Notice that homologous chromosomes are the same size and have the same centromere positions and banding patterns. A human male would have an XY chromosome pair instead of the XX pair. Credit Andreas Bloser et al. In a given species, we can identify chromosomes by their number, size, centromere position, and banding pattern. In a human karyotype, autosomes or body chromosomes, all of the non-sex chromosomes are generally organized in approximate order of size from largest, chromosome 1, to smallest, chromosome 22. The X and Y chromosomes are not autosomes. However, chromosome 21 is actually shorter than chromosome 22. Researchers discovered this after naming Down syndrome as trisomy 21, reflecting how this disorder results from possessing one extra chromosome 21, 3 total. Not wanting to change the name of this important disorder, scientists retain the numbering of chromosome 21 despite describing it having the shortest set of chromosomes. We may designate the chromosome arms projecting from either end of the centromere as short or long, depending on their relative lengths. We abbreviate the short arm P for petite, whereas we abbreviate the long arm Q because it follows P alphabetically. Numbers further subdivide and denote each arm. Using this naming system, we can describe chromosome locations consistently in the scientific literature. Career connection. Geneticists use karyograms to identify chromosomal aberrations. Although we refer to Mendel as the father of modern genetics, he performed his experiments with none of the tools that the geneticists of today routinely employ. One such powerful cytological technique is karyotyping, a method in which geneticists can identify traits characterized by chromosomal abnormalities from a single cell. To observe an individual's karyotype, a geneticist first collects a person's cells, like white blood cells, from a blood sample or other tissue. In the laboratory, the geneticist stimulates the isolated cells to begin actively dividing. The geneticist then applies the chemical colchicine to cells to arrest condensed chromosomes in metaphase. The geneticist then induces swelling in the cells using a hypotonic solution, so the chromosomes spread apart. Finally, the geneticist preserves the sample in a fixative and applies it to a slide. The geneticist then stains chromosomes with one of several dyes to better visualize each chromosome pair's distinct and reproducible banding patterns. Following staining, the geneticist views the chromosomes using bright field microscopy. A common stain choice is the Giemsa stain. Giemsa staining results in approximately 400–800 bands of tightly coiled DNA and condensed proteins arranged along all 23 chromosome pairs. An experienced geneticist can identify each band. In addition to the banding patterns, geneticists further identify chromosomes on the basis of size and centromere location. To obtain the classic depiction of the karyotype, in which homologous chromosome pairs align in numerical order from longest to shortest, the geneticist obtains a digital image, identifies each chromosome, and manually arranges the chromosomes into this pattern, figure 13.5. At its most basic, the karyogram may reveal genetic abnormalities in which an individual has too many or too few chromosomes per cell. Examples of this are Down syndrome, which one identifies by a third copy of chromosome 21, and Turner syndrome, which is characterized by the presence of only one X chromosome in females instead of the normal 2. Geneticists can also identify large DNA deletions or insertions. For instance, geneticists can identify Jacobson syndrome, which involves distinctive facial features as well as heart and bleeding defects, by a deletion on chromosome 11. Finally, the karyotype can pinpoint translocations, which occur when a segment of genetic material breaks from one chromosome and reattaches to another chromosome or to a different part of the same chromosome. Translocations are implicated in certain cancers, including chronic myelogenous leukemia. During Mendel's lifetime, inheritance was an abstract concept that one could only infer by performing crosses and observing the traits that offspring expressed. By observing a karyogram, today's geneticists can actually visualize an individual's chromosomal composition to confirm or predict genetic abnormalities in offspring, even before birth. Chromosome number disorders. Of all of the chromosomal disorders, chromosome number abnormalities are the most obviously identifiable from a karyogram. Chromosome number disorders include duplicating or losing entire chromosomes, as well as changes in the number of complete sets of chromosomes. They are caused by nondisjunction, which occurs when homologous chromosome pairs or sister chromatids fail to separate during meiosis. Misaligned or incomplete synapsis, or a spindle apparatus dysfunction that facilitates chromosome migration, can cause nondisjunction. The risk of nondisjunction occurring increases with the parent's age. Nondisjunction can occur during either meiosis I or II, with differing results, figure 13.6. If homologous chromosomes fail to separate during meiosis I, the result is 2 gametes that lack that particular chromosome and 2 gametes with 2 chromosome copies. If sister chromatids fail to separate during meiosis II, the result is 1 gamete that lacks that chromosome, 2 normal gametes with 1 chromosome copy, and 1 gamete with 2 chromosome copies. Visual connection. This illustration shows nondisjunction that occurs during meiosis I. Nondisjunction during meiosis I occurs when a homologous pair fails to separate and results in 2 gametes with N+1 chromosomes and 2 gametes with N-1 chromosomes. Nondisjunction during meiosis II would occur when sister chromatids fail to separate and results in 1 gamete with N+1 chromosomes, 1 gamete with N-1 chromosomes, and 2 normal gametes. Figure 13. 6. Nondisjunction occurs when homologous chromosomes or sister chromatids fail to separate during meiosis, resulting in an abnormal chromosome number. Nondisjunction may occur during meiosis I or meiosis II. Credit: Rao, A. and Tag, A. Department of Biology, Texas A&M University. Which of the following statements about nondisjunction is true? Nondisjunction only results in gametes with N+1 or N1 chromosomes. Nondisjunction occurring during meiosis II results in 50% normal gametes. Nondisjunction during meiosis I results in 50% normal gametes. Nondisjunction always results in 4 different kinds of gametes. Aneuploidy. Scientists call an individual with the appropriate number of chromosomes for their species euploid. In humans, euploidy corresponds to 22 pairs of autosomes and 1 pair of sex chromosomes. An individual with an error in chromosome number is described as aneuploid, a term that includes monosomy, losing 1 chromosome, or trisomy, gaining an extraneous chromosome. Monosomic human zygotes missing any 1 copy of an autosome invariably fail to develop to birth because they lack essential genes. This underscores the importance of gene dosage in humans. Most autosomal trisomies also fail to develop to birth. However, duplications of some smaller chromosomes, 13, 15, 18, 21, or 22, can result in offspring that survive for several weeks to many years. Trisomic individuals suffer from a different type of genetic imbalance, an excess in gene dose. Individuals with an extra chromosome may synthesize an abundance of the gene products, which that chromosome encodes. This extra dose, 150% of specific genes, can lead to a number of functional challenges and often precludes development. The most common trisomy among viable births is that of chromosome 21, which corresponds to Down syndrome. Short stature and stunted digits. Facial distinctions that include a broad skull and large tongue, and significant developmental delays characterize individuals with this inherited disorder. We can correlate the incidence of Down syndrome with maternal age. Older people are more likely to become pregnant with fetuses carrying the trisomy 21 genotype, figure 13.7. This graph shows the risk of Down syndrome in the fetus with increasing maternal age. Risk dramatically increases past a maternal age of 35. Figure 13. 7. The incidence of having a fetus with trisomy 21 increases dramatically with maternal age. Link to learning. Visualize adding a chromosome that leads to Down syndrome in this video simulation. Polyploidy. We call an individual with more than the correct number of chromosome sets. 2. For diploid species, polyploid. For instance, fertilizing an abnormal diploid egg with a normal haploid sperm would yield a triploid zygote. Polyploid animals are extremely rare, with only a few examples among the flatworms, crustaceans, amphibians, fish, and lizards. Polyploid animals are sterile because meiosis cannot proceed normally and instead produces mostly aneuploid daughter cells that cannot yield viable zygotes. Rarely, polyploid animals can reproduce asexually by haplodiploidy, in which an unfertilized egg divides mitotically to produce offspring. In contrast, polyploidy is very common in the plant kingdom, and polyploid plants tend to be larger and more robust than euploids of their species, figure 13. 8. Photo shows an orange day lily, which is a plant with a large flower. The flower looks like it is bursting with orange petals. Figure 13. 8. As with many polyploid plants, this triploid orange daylily, Hemerocallis fulva, is particularly large and robust, and grows flowers with triple the number of petals of its diploid counterparts. Credit Steve Karg. Sex chromosome nondisjunction in humans. Humans display dramatic deleterious effects with autosomal trisomies and monosomies. Therefore, it may seem counterintuitive that human females and males can function normally, despite carrying different numbers of the X chromosome. Rather than a gain or loss of autosomes, variations in the number of sex chromosomes occur with relatively mild effects. In part, this happens because of the molecular process X inactivation. Early in development, when female mammalian embryos consist of just a few thousand cells, relative to trillions in the newborn, one X chromosome in each cell inactivates by tightly condensing into a quiescent, dormant structure or a bar body. The chance that an X chromosome, maternally or paternally derived, inactivates in each cell is random. But once this occurs, all cells derived from that one will have the same inactive X chromosome or bar body. By this process, females compensate for their double genetic dose of X chromosome. In so-called tortoiseshell cats, we observe embryonic X inactivation as color variegation, figure 13.9. Females that are heterozygous for an X-linked coat color gene will express one of two different coat colors over different regions of their body, corresponding to whichever X chromosome inactivates in that region's embryonic cell progenitor. Photo shows a tortoiseshell cat with orange and black fur. Figure 13. 9. In cats, the gene for coat color is located on the X chromosome. In female cats' embryonic development, one of the two X chromosomes randomly inactivates in each cell, resulting in a tortoiseshell pattern if the cat has two different alleles for coat color. Male cats having only one X chromosome never exhibit a tortoiseshell coat color. Credit Michael Bodega. An individual carrying an abnormal number of X chromosomes will inactivate all but one X chromosome in each of her cells. However, even inactivated X chromosomes continue to express a few genes, and X chromosomes must reactivate for the proper maturation of female ovaries. As a result, X chromosomal abnormalities typically occur with mild intellectual and physical disorders or disabilities, as well as sterility. If the X chromosome is absent altogether, the individual will not develop in utero. Scientists have identified and characterized several errors in sex chromosome number. Individuals with 3 X chromosomes, triplo X, are phenotypically female but express developmental delays and reduced fertility. The XXY genotype, corresponding to one type of Klinefelter syndrome, corresponds to phenotypically male individuals with small testes, enlarged breasts, and reduced body hair. More complex types of Klinefelter syndrome exist in which the individual has as many as 5 X chromosomes. In all types, every X chromosome except one undergoes inactivation to compensate for the excess genetic dosage. We see this as several bar bodies in each cell nucleus. Turner syndrome, characterized as an X0 genotype, i.e., only a single sex chromosome, corresponds to a phenotypically female individual with short stature, webbed skin in the neck region, hearing and cardiac impairments, and sterility. Duplications and deletions. In addition to losing or gaining an entire chromosome, a chromosomal segment may duplicate or lose itself. Duplications and deletions often produce offspring that survive but exhibit abnormalities. Duplicated chromosomal segments may fuse to existing chromosomes or may be free in the nucleus. Cry-du-chat, from the French for cry of the cat, is a syndrome that occurs with nervous system abnormalities and identifiable physical features that result from a deletion of most 5p, the small arm of chromosome 5. Figure 13. 10. Infants with this genotype emit a characteristic high-pitched cry on which the disorder's name is based. Photos show a boy with cry-du-chat syndrome. In parts A, B, C, and D of the image, he is 2, 4, 9, and 12 years of age, respectively. Figure 13. 10. This figure shows an individual with cry-du-chat syndrome at 2, 4, 9, and 12 years of age. Credit, Paula Maynardi. Chromosomal structural rearrangements. Cytologists have characterized numerous structural rearrangements in chromosomes, but chromosome inversions and translocations are the most common. We can identify both during meiosis by the adaptive pairing of rearranged chromosomes with their former homologs to maintain appropriate gene alignment. If the genes on two homologs are not oriented correctly, a recombination event could result in losing genes from one chromosome and gaining genes on the other. This would produce aneuploid gametes, chromosome inversions. A chromosome inversion is the detachment, 180 degrees rotation, and reinsertion of part of a chromosome. Inversions may occur in nature as a result of mechanical shear, or from transposable elements action. Special DNA sequences capable of facilitating rearranging chromosome segments with the help of enzymes that cut and paste DNA sequences. Unless they disrupt a gene sequence, inversions only change gene orientation and are likely to have more mild effects than aneuploid errors. However, altered gene orientation can result in functional changes because regulators of gene expression could move out of position with respect to their targets, causing aberrant levels of gene products. An inversion can be paracentric and include the centromere, or paracentric and occur outside the centromere, figure 1311. A paracentric inversion that is asymmetric about the centromere can change the chromosome arm's relative lengths, making these inversions easily identifiable. Illustration shows paracentric and paracentric inversions. In this example, the order of genes in the normal chromosome is a B, C, D, E, F, with the centromere between genes C and D. In the paracentric inversion, the order is a B, D, C, E, F in the paracentric inversion example. The resulting gene order is a B, C, D, F, E, figure 13. 11 paracentric inversions include the centromere, and paracentric inversions do not. A paracentric inversion can change the chromosome arm's relative lengths. A paracentric inversion cannot. When one homologous chromosome undergoes an inversion but the other does not, the individual is an inversion heterozygote. To maintain point-for-point synapsis during meiosis, one homolog must form a loop, and the other homolog must mold around it. Although this topology can ensure that the genes correctly align, it also forces the homologs to stretch and can occur with imprecise synapsis regions. Figure 1312. This illustration shows the inversion pairing that occurs when one chromosome undergoes inversion, but the other does not. For chromosome alignment to occur during meiosis, one chromosome must form an inverted loop while the other conforms around it. Figure 13. 12. When one chromosome undergoes an inversion, but the other does not, one chromosome must form an inverted loop to retain point-for-point interaction during synapsis. This inversion pairing is essential to maintaining gene alignment during meiosis and to allow for recombination. Evolution connection. The chromosome 18 inversion. Not all chromosomes' structural rearrangements produce non-viable, impaired, or infertile individuals. In rare instances, such a change can result in new species evolving. In fact, a paracentric inversion in chromosome 18 appears to have contributed to human evolution. This inversion is not present in our closest genetic relatives, the chimpanzees. Humans and chimpanzees differ cytogenetically by paracentric inversions on several chromosomes and by the fusion of two separate chromosomes in chimpanzees that correspond to chromosome 2 in humans. Scientists believe the paracentric chromosome 18 inversion occurred in early humans following their divergence from a common ancestor, with chimpanzees approximately 5 million years ago. Researchers characterizing this inversion have suggested that approximately 19,000 nucleotide bases were duplicated on 18p, and the duplicated region inverted and reinserted on chromosome 18 of an ancestral human. A comparison of human and chimpanzee genes in the region of this inversion indicates that two genes, ROC1 and USP14, that are adjacent on chimpanzee chromosome 17, which corresponds to human chromosome 18, are more distantly positioned on human chromosome 18. This suggests that one of the inversion breakpoints occurred between these two genes. Interestingly, humans and chimpanzees express USP14 at distinct levels in specific cell types, including cortical cells and fibroblasts. Perhaps the chromosome 18 inversion in an ancestral human repositioned specific genes and reset their expression levels in a useful way. Because both ROC1 and USP14 encode cellular enzymes, a change in their expression could alter cellular function. We do not know how this inversion contributed to hominid evolution, but it appears to be a significant factor in the divergence of humans from other primates. 1. Translocations. A translocation occurs when a chromosome segment dissociates and reattaches to a different non-homologous chromosome. Translocations can be benign or have devastating effects depending on how the positions of genes are altered with respect to regulatory sequences. Notably, specific translocations have occurred with several cancers and with schizophrenia. Reciprocal translocations result from exchanging chromosome segments between two non-homologous chromosomes, such that there is no genetic information gain or loss. Figure 1313. Illustration shows a reciprocal translocation in which DNA is transferred from one chromosome to another. Some DNA is also transferred from the second chromosome back to the first. No genetic information is gained or lost in the process. Figure 13. 13. A reciprocal translocation occurs when a DNA segment transfers from one chromosome to another non-homologous chromosome. Credit: Modification of work by National Human Genome Research, USA.